Hello, I would like to tell you today about the direct antioxidant effect of phenolic phytochemicals on the intestinal mucosa, and I would like to focus the attention in particular on hydroxytyrosol. The gastrointestinal tract is exposed to many pro-oxidant conditions arising from both endogenous and exogenous routes. An important exogenous route is represented by the diet. For example, some reactive species are present in the food. For example, lipid peroxide, aldehydes, hydrogen peroxide, or M-protein. Or we have to consider also the presence of pollutants contaminating food. Other reactive species arise from chemical reactions of dietary components in the low pH of stomach. Moreover, some diet-derived bacteria and toxins are able to increase the ROS production by the activation of the immune cells naturally present in the gastrointestinal tract. In addition to the exogenous routes of the exposure to pro-oxidant conditions, we have to consider reactive species released by the activity of some enzymes highly expressed in the intestine, such as NADPH oxidase and xanthine oxidase. So if we consider the important function of the gastrointestinal epithelium as an interface between the internal and the external environment of the organism, and if we consider the variety of exposure conditions to pro-oxidant conditions, we can understand that the maintenance of a correct redox balance in the gastrointestinal tract is of pivotal importance for the health status of the organism. So, the excess of reactive species under anomalous conditions and is able to contribute to the progress of disease in the gastrointestinal tract. And among the segments of the gastrointestinal tract, the colon is one of the segments more exposed to pro-oxidant conditions. The reasons can be found for example, in the longer exposure to oxidized food particles, toxins, and redox active minerals because of the prolonged transit time of the bolus in this district. Moreover, we have also to consider bacterial microflora, which generates a consistent quantity of reactive metabolites. And some authors underline the fact that the pro-oxidant environment of the colon compared to other segments of the gastrointestinal tract, such as, for example, the small intestine, may contribute to the greater cancer susceptibility of this district. So the maintenance of the correct gastrointestinal redox balance through the involvement of endogenous and exogenous antioxidant defenses mainly derived from the diet is an important aspect to be considered for the protection of human health. Plant polyphenols represent the most abundant antioxidants in the diet and contribute to the exogenous antioxidant defenses of the organism. In particular, phenolic compounds typically included in the Mediterranean diet are of great interest for their health benefits described in literature, their use as dietary supplements, and their use as functional ingredients. But the bioavailability of polyphenolic compound is a certain way to debate it, because this compound, after absorption, undergo the hepatic biotransformation, which can produce sometimes metabolites which are less effective with respect to the parental compound. On the other hand, the gastrointestinal epithelium is exposed to this compound after ingestion directly before the hepatic biotransformation. Moreover, polyphenol concentration in the gastrointestinal tract lumen is able to reach concentration up to several hundred micromolar, and the highest polyphenol concentration are observed in the colon. In particular, hydroxytyrosol is a phenolic compound occurring in olive leaves and pulp and typically present in olive oil, which is the main fat source in the Mediterranean diet. It is one of the most potent antioxidants from nature as assessed by in vitro studies, as you can see by the analysis of its ORAC activity in vitro. Hydroxytyrosol has been recognized to protect low-density lipoprotein from oxidative damage by the European Food Safety Authority. Hydroxytyrosol has been demonstrated to protect cells against hydrogen peroxide-induced DNA damage, and moreover, its anti-inflammatory activity and anti-cancer effects are reported in literature. After ingestion, the hydroxytyrosol during its transit along the gastrointestinal tract is able to maintain its stability in the gastric juice at the duodenal in the duodenal fluid. In the stomach, the hydroxytyrosol concentration is increased by the hydrolysis of its precursors, represented by oleuropein and lymphocytes, which are not absorbed at the gastric level. And from about 40 to 95% of ingested hydroxytyrosol is absorbed by the small intestine epithelium. Hydroxytyrosol not absorbed in the intestine level accumulates in the colon, where it is stable with respect to the microflora action. And microflora release further hydroxytyrosol from not absorbed oleuropein. All these aspects contribute to the fact that hydroxytyrosol in the colon is present in concentration included in the high micromolar range. So the colon epithelium represents one of the tissues of the organism exposed to the higher concentration of this compound before its hepatic biodegradation. So it is very interesting to assess its direct effect on the intestinal mucosa, its direct antioxidant effect. So the objective of the research is to detect the absorbability of hydroxytyrosol in rat colon surface cells. To assess its intracellular antioxidant activity in the colon epithelium, and to assess its contribution to the protection of the intestinal epithelium from pro-oxidant conditions. The experimental model utilized in our study was represented by freshly isolated rat colon explants, which closely resembled the physiological and morphological characteristics of the colon epithelium in vivo. And we applied in situ confocal microscopy on colon explants charged with the ROS-sensitive cell permeant dye CM H₂DCF-DA. Here you can see the typical appearance of the surface of colon epithelium under microscopic bright field conditions of observation. You can see superficial colonocyte arranged around a central crypt. And when we observed the surface of the tissue under confocal microscopy, we focused our attention on both basal ROS fluorescence, produced by the cell, and on the fluorescence of the colonocyte after induced by the exposure to an oxidative challenge, represented by the exposure to hydrogen peroxide after 15 minutes of exposure. It is what you can see in the graph, in the image on the in the picture on the right. So we utilized hydrogen peroxide as oxidative challenge because hydrogen peroxide is a physiological actor, oxidative actor in the colon, because it is normally produced by bacteria and phagocyte cells under inflammation. So it is a pro-oxidant agent important for the progression of inflammatory pathologies in the colon. And the concentration utilized in our study, 400 micromolar on the base of dose response previous studies, is included in the normal physiological concentration that we can find in the colon epithelium under inflammatory conditions. Preliminarily, we wanted to assess if in our experimental conditions, the experimental exposure of the explants to hydroxytyrosol was able in a certain way to alter the ion transport properties and permeability of rat colon epithelium. So we carried out a using chamber experiment in which the explants were exposed to hydroxytyrosol, to 100 micromolar hydroxytyrosol, one of the highest concentration tested in our study, and the incubation was prolonged for an hour. As you can see, the pre-exposure of the tissue with hydroxytyrosol did not produce any significant changes in transepithelial potential, short-circuit current, and transepithelial resistance of the colon, suggesting that the experimental exposure of the explants to hydroxytyrosol does not interfere with the functional transport properties of the epithelium itself. So first, we wanted to assess the effect of hydroxytyrosol on the basal ROS production in surfaceal colonocyte, and our experimental design included control explants incubated for an hour with PPS, washed, incubated with a probe, washed again, and observed under confocal microscopy, and treated explants which were pre-incubated for an hour with hydroxytyrosol at different concentration and the concentration tested ranged from 10 micromolar to 200 micromolar. As you can see in the graph on the left, we measured the percentage of residual basal fluorescence in the cell, and the incubation with hydroxytyrosol produced a dose-dependent inhibition in the basal fluorescence in the ROS basal fluorescence of colonocyte. These results suggest that after an hour incubation, hydroxytyrosol reaches intracellular concentration suitable to exert a significant dose-dependent reduction of the basal ROS generation in colonocyte. And these indicate a possible preventive role in oxidative stress associated condition of this compound in the colon. Then we wanted to assess the effect of hydroxytyrosol against an exogenous oxidative challenge induced by the exposure to hydrogen peroxide. So our experimental design included the control explant incubated for an hour with PPS, washed, exposed to an oxidative challenge represented by the exposure to hydrogen peroxide 400 micromolar for 50 minutes. The explants were washed again and charged with the probe and then observed, and treated explants that were pre-incubated for an hour with hydroxytyrosol at a different concentration. we observed a significant dose-dependent inhibition of the hydrogen peroxide induced fluorescence in colonocyte. So these results suggest that hydroxytyrosol induced a dose-dependent antioxidant action directly on the colon mucosa exposed to an hydrogen peroxide challenge. And this suggests that hydrogen peroxide directly contribute to the protection of the intestinal epithelium from pro-oxidant conditions. Then we wanted to compare the antioxidant effect exerted by hydroxytyrosol against an oxidative challenge with the effect exerted in the same experimental conditions by Trolox, which is a synthetic analog of vitamin E and widely utilized as a standard in the in vitro assay of the antioxidant activity. As you can see, the inhibitory effect exerted by the exposure, the pre-incubation with 50 micromolar hydroxytyrosol, the inhibitory effect on the hydrogen peroxide-induced fluorescence, was comparable to the effect produced by the pre-incubation with 10 micromolar Trolox. Finally, we wanted to assess if the antioxidant activity of hydroxytyrosol and the antioxidant protective effect exerted by hydroxytyrosol on the colon mucosa against an oxidative challenge was also exerted against an oxidative challenge produced by another pro-oxidant agent, such as terbutyl hydroperoxide. The choice of this compound comes from the fact that terbutyl hydroperoxide is a synthetic lipid peroxide widely utilized as a model molecule in the studies assessing the damaging effect of lipid peroxidation. As you can see in the upper graph, the oxidative challenge produced on the colon epithelium by terbutyl hydroperoxide is quite similar to the oxidative challenge induced by hydrogen peroxide expressed in cell fluorescence intensity. And when the tissues, the explants, were pre-incubated with hydroxytyrosol, the inhibitory effect exerted by hydroxytyrosol on the oxidative challenge induced by terbutyl hydroperoxide, 100 micromolar, was more dramatic with respect to the effect exerted against an oxidative challenge produced by hydrogen peroxide. So this result suggests that the oxidative challenge exerted by these two agents may involve different oxidative reactions and different sites whose sensitivity to hydroxytyrosol scavenging activity is different. So in conclusion, our study indicates that hydroxytyrosol directly contributes to the redox balance of the colon epithelium by reducing ROS both in basal conditions and in pro-oxidant conditions, and potentially protects the mucosal epithelium from oxidative stress-related damage. In perspective, our results support the potential of hydroxytyrosol as a functional food ingredient with applications in the protection of intestinal mucosa against oxidative challenge. And these results open to further studies involving hydroxytyrosol aimed at the prevention and treatment of pathological conditions associated with the excessive exposure to oxidative stress in the gastrointestinal tract, in particular on the colon epithelium, which is particularly exposed to pro-oxidant conditions in both physiological and pathophysiological conditions and prone to cancer susceptibility. Thank you for your attention.